everyone, my name is Hanna and today I'm going to be discussing gene editing in farm animals. In this presentation, I will discuss how targeted gene edits can reduce disease and improve welfare, but only if controlled with strong restrictions and safety procedures. Throughout this presentation, I will cover what gene editing is, 3 animal case studies of examples, the controversies, ethics, policies and risk controls, and finally conclusions and takeaways. Firstly, let's start out by talking about how gene editing works. In order to do this, a guide RNA finds the spot of the gene that wants to be edited, and the Cas enzyme cuts the DNA. The cell then repairs the cut, and the small targeted change comes to life. Now that you've gotten a little glimpse into the biology behind it, I'm going to explain what gene editing it is within itself. So the definition of gene editing is precise DNA changes. This can be insertions, which is additions, deletions, or substitutions where you switch out a gene. The main tool used in this procedure is a CRISPR. They're tiny scissors that are guided to one DNA spot. In terms of the FDA, heritable intentional genomic alterations, also known as IGAs, are what editing is referred as. It is used to edit embryos to breed into the trait into herds or flocks. Why would we use editing versus breeding? Because it is faster and it's also more accurate for single well-understood traits. Now let's go through the timelining of the editing. So firstly, we would edit the embryos, and then we would have to screen for the right edit. And then after that, when the edit is implanted, you raise the animal, because remember, we edit in the embryonic stage, not while they're alive. And then you would continue to breed this into the bloodline and monitor the edit as time goes on to report any successes or challenges. That way it can be altered. The first edited gene study case I'm going to talk about is related to pigs, and the target of this edit is the CDL63 receptor. This receptor edit blocks the virus from entering, and the virus is PRRS, which is a respiratory disease, and it causes late-term abortions and stillbirths in sows, which are the mother pigs, and it's a respiratory disease in piglets, and it's the most costly swine disease in pigs. So if you have this expensive disease that's affecting pigs, then you take this edit. It allows for the disease to not be as prominent and be even resistant, possibly. And then the FDA has approved the PRRS resistant pig gene edit this year in 2025, and a regulatory pathway has been officially defined. Another interesting trait of this specific case study is that it proves that multiple gene edits can coexist at the same time and also improve resilience without harming the animal. This is because we can take the gene edit that we talked about before, and you can add another gene edit, which is the PAPN, and they call this the double knockout, and it provides resistance to not only the PRRS, but also another disease, and it also reduces susceptibility to a whole other disease as well, which is very interesting. The next case study of edited genes that I would like to talk about takes place in chickens, and it's the avian influenza or the bird flu resistance, and it happens in the ANP32A edit. It's a host factor that's used by the flu polymerase. This edit changes the way that the flu affects the animal, and it slows it down. This is because it would reduce the bird flu outbreaks, and it would also cultivate economic benefits, considering it would be less sick birds that we would have to then tend for. The study has shown that it works well for normal doses. However, it's not perfect in large doses. The result of the study is that 9 out of the 10 edited birds resisted the normal dose. However, high dose exposures cause breakthroughs, showing that this is a promising edit, but it's not yet perfect or approved by the FDA. The next case study of why we should edit genes or not takes place in cows. The first one I would like to talk about is heat tolerance, or the slick coat. The target of this is the PRLR, also known as the slick variant, which causes short, sleek hair, which helps with heat dispersion, which allows for temperature regulation of the animal. The FDA has also determined that this is a low-risk edit, and it does in fact produce slick edited cattle. The next one I would like to talk about is the hornless polled dairy cattle, which involves the calves being born without horns, which avoids the dehorning process, which is painful for the animal and traumatic. The target of this gene is the polled, or PC Celtic allele, and due to the editing, the calves then become hornless, which takes away that whole procedure. Okay, and then the final cow-related gene edit I would like to talk about is the bovine tuberculosis resistance, also known as the NRAMP1. The target is the NRAMP1, and it's a natural resistance gene, which usually in FDA standards means it's lower risk. And this edit increases the resistance of tuberculosis in edited cattle, but it's still in the research stage. And then one more edit I actually want to talk about is the hyperallergenic milk. This one is very, very interesting to me. The target of this is the beta-lactin gene, which is the major milk allergen in cow milk. So the edited cattle can produce milk with the no beta-lactin. Therefore, it could reduce possible allergies, which is very interesting. The final case study I would like to talk about is more of a controversial, unethical question mark kind of situation. It involves the myostatin MSTN edit, which is used to increase muscle and feed efficiency. It gives the animal a higher body weight, which in turn improves the meat quality. It also transforms the feed into muscle faster, which would potentially lower production cost and help with, you would have to pay less for feed for the animals because it's performing the muscle faster. This is a powerful edit, but it also can create welfare trade-offs. As I talked about, it's ethical question marks. It has high potential as a genetic edit, but it also poses the question of where do we draw the line of making the edit potentially just for the human benefit and not for the quality of life for the animal. Now I would like to talk about the benefits that can be seen in farming. So the first one is that it can be less contagious disease losses, as we've seen in the respiratory and miscarriage in swines, as well as the avian flu. Also, it can improve animal welfare, like removing the painful procedures such as dehorning. It also can help with food security and climate resilience, healthier animals, and the better heat tolerance due to the slick gene. And it's also faster than selective breeding, especially when you're looking at one or few gene traits at once. Now I'm going to move into the benefits for humans, and this would be seen in public health due to lower antibiotic use on farms, which also supports the antimicrobial resistance efforts. And then consumer benefits, we see more stable prices and availability. Also, the potential allergy-friendly options due to the milk allergen, BLG, free milk in cows. We would also see worker and community benefits due to fewer high-risk procedures like dehorning. It would be safer handling for not only farm owners but the animals themselves, and it could also have fewer outbreak holes. Also, for climate resistance due to the heat tolerant cattle, which helps stabilize supply during hotter summers, and also benefits the economy due to reduced disease losses, and it improves farm viability and regional employment stability. Now that we've looked at some case studies and some benefits, I'm going to now move into the risks. And the first risk I would like to talk about involves the biology and tech. So, firstly, off target or extra changes. It can sometimes the edit can land in the wrong spot, or even it could end up in the right spot but changes more than it was expected to. This matters because that could subtly affect other traits that aren't supposed to be affected, which should cause a whole spew of problems. In order to fix this, we need to do whole genome checks and confirm that the exact DNA structure that is being targeted is the correct edited site. Another risk is mixed in cells in the founders. So the first edited animal can have a mix of some cells that do get the edit and some that do not. And this matters because the trait can be weaker or less consistent in babies following, which defeats the point of the edit. And finally, extra embryo losses. Gene editing can sometimes lower embryo survival compared to normal breeding, and this matters because it's a loss of life of the embryo. It would be less cattle, so that would affect not just cattle, all animals that are being edited, and that would affect not only the welfare, but it would be inefficient because then you'd be losing animals. That could also affect the economy. In order to prevent this, perfect lab steps need to be taken, and you need to compare outcomes with the unedited controls in order to find the best procedures. The next risk I would like to talk about involves the animal welfare, and this one's a very big morally important question of whether gene editing should be allowed. And the first point is that some edits can put strain on the body. Edits that push extreme growth or muscle or milk can overload the animal, such as the edit that we talked about previously that increases muscle mass of the animal, which is the double muscling, like the STN edits, which makes the cows very large, but it also is more beneficial to humans than the animal. It could cause a spew of problems in the animals, such as more C-sections, loss of life in calves, and mobility problems in their walking due to increased muscle. In order to combat this, we would have to track welfare and not just care about the output of the gene. We have to ensure that the animal has a good quality of life, not that the edit was made successfully, because it still has to be ethically correct in order for the gene edit to be approved. The third risk I would like to talk about involves ecology and the markets. This was because it would be less genetic diversity within animals. So if one edited line takes over an animal population, then the herds can become too similar. And this matters because if a new disease that this edit is susceptible to, it could wipe out a lot of animals at once. So in order to combat this, we need to make sure to keep multiple edited lines, but also keep conventional genetics in the mix. There's also different rules across all the different countries. This matters because it affects exports and market access. It also affects timing. I know the US is a little bit more forward on this than compared to the UK, who is still only looking to do this in plants as of now. They are looking to eventually move into animals, but right now they haven't gotten there yet. So it would affect trade. And then A clear gene-edited labels need to be provided because people want to know what they're buying and why it was edited, which allows them to make an educated choice on the matter. It also is linked to, we need to have access to the safety data as well to make the best choice in the approval. The fourth and final risk I want to touch on is the antibiotic claims. The edits that boost disease or heat resilience can in turn mean fewer infections, which means less antibiotic use. However, animals will still get sick sometimes. That's inevitable, so there still needs to be vaccines, biosecurity, diagnostics, and vet oversight in place in order to check on the welfare of the animal first. Speaking of welfare of the animals, it's very important to touch on whether or not the edit hurts the animals and if it's humane. However, the edit does occur at the embryonic or zygote stage, which in turn means it's not painful to the animal once it is born. But the focus needs to be on trait consequences and the methods used during development to always ensure the welfare of the animal first, and that the gene edit isn't causing new consequences or affecting other consequences. In terms of humanity, it does reduce the suffering in terms of disease, dehorning, and heat stress. And with strong welfare oversight and transparent risk testing, it could be very beneficial. When it gets to being not humane is when it adds pain or harm or even chronic stress to the animal, such as the extreme growth traits, and there's no safeguards, especially if there's poor surrogate and newborn welfare. There needs to be a lot of oversight when it comes to that. But as I've mentioned before, that growth trait in total is a humanity question of ethics. So now I'm going to move into the moving parts of the operations and specific of who does what. Firstly, there's the breeders who validate the traits and make sure the gene edit works. They also need to check the performance of the edit and on the welfare of the animals. And then we move into the regulators who review the safety, the risks, and the labels, and make sure that everything is clear and that safety standards are in check. And then they can either approve or not of the gene edit. Then we move down to farmers and vets, whose job is to monitor the herds and report any problems and fix them as soon as possible, and then care for the animals. And then we get to the public, who have the ability to make an informed choice based on clear labels and access to the safety data. Now we're going to move into the rules and regulations that matter and are put in place. In the US, currently we use a medical style review, such as the FDA, and it's all risk-based. And we have learned that natural-like edits can be determined as lower risk. So if an edit matches a variant that's already found in nature, the risk is considered to be lower, usually. However, that's just in the US. We know that elsewhere rules are still evolving. As I've mentioned, in the UK, they're looking to start doing gene edits on plants, but have not yet moved into animals. Some risk controls that help to combat against the risks that I've previously mentioned are before market is deep checks, as I mentioned, full genome checks, to ensure that the gene trait change is correct, efficient, and safe, and beneficial, not harmful to the animal. After market, we need to track our outcomes, again, to make sure that everything is working out as it's supposed to, and if there are problems, to report them so that we can figure out a solution as fast as possible to get back on track or even make it better. Finally, my conclusions based on my findings are that there are real positives and gains that are possible due to gene editing. However, the welfare of the animal and fairness are key. We cannot do edits if it harms the animal, or even if small farms who don't have access to enough funds or resources fall behind in the process and are then in turn hurting our farmers, and that there absolutely needs to be strong oversight in order for this to be safe. We need clear rules, strong monitoring, reporting, and fixing any problems that do occur. Finally, I'm going to leave you with some takeaway questions. Firstly, being is if you think that overall gene editing is humane and ethical. Secondly, do you think it's okay to do a gene edit if it's more beneficial to humans than the animals? One example of this is the muscle growth edit, which is more important after the death of the cow in order for better meat production. And finally, if there were clear labels and reasons as to why the edit was made that were made clear to you, do you think as a consumer you would feel comfortable? Thank you.